Imagine a world where extinct creatures roam the earth again. Picture woolly mammoths traversing the frozen tundra, their colossal forms casting long shadows across the ice. Imagine flocks of passenger pigeons darkening the skies, their numbers so vast they blot out the sun. This is the tantalizing promise of de-extinction, the audacious scientific pursuit of bringing vanished species back from oblivion. For centuries, extinction has been considered an irreversible full stop in the story of a species. Yet, with the advent of groundbreaking genetic technologies, the possibility of turning back the clock on extinction is suddenly within our grasp. We are on the cusp of an era where we might have the power to rewrite the very narrative of life on Earth. But should we? The prospect of resurrecting extinct creatures sparks both awe and apprehension. It begs us to confront fundamental questions about our role in the natural world. Do we have the right to bring back species that we ourselves drove to extinction? And what consequences await both these resurrected creatures and our planet if we dare to open this Pandora's box of genetic possibilities? De-extinction is not simply the stuff of science fiction, it is rapidly becoming a scientific reality. At its core, it involves manipulating the very building blocks of life, DNA, to recreate extinct organisms. Scientists are exploring several techniques, each with its own set of promises and limitations. Cloning represents the most direct approach. This technique involves extracting the nucleus, which contains the complete genetic blueprint from a preserved cell of the extinct animal. This nucleus is then inserted into an egg cell from a closely related living species, which acts as a surrogate. If successful, the egg develops into an embryo carrying the genetic legacy of the extinct species. Gene editing offers a more nuanced approach. This revolutionary technology, often using tools like CRISPR-Cas9, allows scientists to precisely target and modify specific genes within the genome of a living species. By strategically editing these genes to resemble those of the extinct relative, scientists can essentially rewrite the evolutionary path, coaxing forth traits that have long been lost. Selective breeding, a more traditional method, also plays a role. By identifying individuals within a living population that possess ancestral traits reminiscent of the extinct species, scientists can selectively breed these individuals over generations. This careful selection process aims to amplify the desired traits, gradually reviving the extinct form within the living population. Each of these methods presents significant challenges, from the technical complexities of manipulating fragile ancient DNA to the ethical considerations of using living animals as surrogates in de-extinction experiments. However, the rapid pace of progress in genetics fuels optimism that these hurdles may soon be overcome. The prospect of de-extinction has sparked a flurry of research, with scientists around the world setting their sights on bringing back a variety of lost creatures. Among the most iconic candidates is the woolly mammoth, a behemoth that once roamed the icy plains of the northern hemisphere. Well-preserved remains frozen in permafrost offer a treasure trove of genetic material, making the mammoth a prime candidate for cloning or gene editing. Another contender is the passenger pigeon, a bird that once darkened the skies of North America with its vast flocks. Though hunted to extinction in the early 20th century, preserved specimens provide valuable DNA samples. Scientists hope to use gene editing to resurrect the passenger pigeon, potentially releasing flocks into the wild to fulfill their ecological role once more. The thylacine, a marsupial predator also known as the Tasmanian tiger, is another compelling candidate. Declared extinct in the 1930s, the thylacine holds a unique place in Australia's ecosystem. Efforts are underway to use gene editing techniques to rewrite the genome of a related species, the dunart, to resemble that of the thylacine, effectively bringing back this iconic predator from the brink. Here, these species represent just a fraction of the creatures that could potentially be revived. From the dodo to the gastric brooding frog, the list of candidates grows longer as scientists delve deeper into the possibilities offered by de-extinction technologies. While the prospect of de-extinction is exhilarating, the path to resurrection is paved with formidable obstacles. One of the most significant challenges lies in the fragile nature of ancient DNA. Over time, DNA degrades, breaking down into smaller and smaller fragments. This degradation makes it incredibly difficult to piece together the complete genetic blueprint of an extinct creature. Even if scientists can overcome the hurdle of DNA degradation, finding suitable surrogates for extinct species poses another major challenge. Cloning requires a closely related living species to act as an egg donor and surrogate mother. However, for many extinct animals, finding a suitable surrogate with compatible reproductive systems and gestation periods is a daunting task. Habitat loss presents a further obstacle. Many extinct animals vanished because their environments were destroyed. Before even considering resurrection, we must ensure that these revived creatures would have a safe and suitable habitat to call home. Reintroducing a species into an environment that can no longer support them would be both irresponsible and ultimately futile. Beyond these practical challenges lie ethical concerns. Some argue that de-extinction is an act of hubris, a dangerous tampering with the natural order. Others worry that focusing on bringing back extinct creatures distracts from the urgent need to protect endangered species currently on the brink. The ethical debate surrounding de-extinction is complex and multifaceted. There are no easy answers, and the decision of whether or not to pursue this technology is fraught with moral and philosophical dilemmas. If you enjoyed this video, be sure to give our channel a subscribe and check out some of our other videos.